Hello, this is Mr. Brady. For today, we are going to be talking about the structure and function of DNA, the molecule that makes us who we are. In order to do this, you will need to make sure that you have a copy of this worksheet for this presentation. It will go in order, so please follow along and pay close attention to what's written here. We're going to start with a short video on the structure of DNA.
All cells require some form of instructions to be able to function properly. They need guidelines, rules, codes for making materials in the cell. And that code is DNA. DNA stands for deoxyribonucleic acid. D for the name of the sugar, N and A for nucleic acid. DNA contains the information that determines inherited characteristics. It has the code for making proteins. DNA is found in the nucleus of eukaryotic cells and in the cytosol of prokaryotes. If we take a closer look at the chromatin inside the nucleus, we can see the structure of the DNA. The DNA has repeating subunits, and those subunits are called monomers, or nucleotides specifically. The nucleotide has three main parts: a phosphate group, a sugar, and a nitrogen base. In DNA, the name of the sugar is deoxyribose, which is part of DNA's name, and there are 4 nitrogen bases in DNA. The bases are thymine, adenine, cytosine, and guanine. 2 of the bases are purines, which have a 2-ring structure, and 2 bases are pyrimidines, which have a 1-ring structure. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. I remember the pyrimidines are the bases with a Y in their name, just like pyrimidine has a Y in its name. A purine always pairs with a pyrimidine, and the slanted shape of the DNA molecule causes it to form a spiral or helix. Because DNA is double-stranded, we use the phrase double helix to describe its structure. There are 4 scientists who are credited with discovering the shape of DNA, and they are Watson, Crick, Wilkins, and Franklin. All of them received a Nobel Prize for their work, except for Rosalind Franklin. She died before the prize was given. Each of these scientists played a role in piecing together the structure of DNA. They learned that along the sides of the molecule was a backbone made of alternating sugar and phosphate molecules. On the inside, like the rungs of a ladder, are the nitrogen bases. Adenine and thymine form hydrogen bonds together. Cytosine and guanine form hydrogen bonds together. To help me remember which bases link together, I think of writing the letters. A and T both use straight lines. C and G use curved lines. I also know that A and T have 2 hydrogen bonds, but C and G have 3 hydrogen bonds by saying AT2, CG3. Silly things like this are actually a great memory tool. Strands of DNA are said to be complementary to one another because A will always be with T and C will always be with G, based on the number of hydrogen bonds that they want to make. You can predict the complementary strand if you know the other strand. So let's use all of that knowledge about DNA and see if we can identify the parts of this blank DNA molecule. The easiest to label are the deoxyribose sugars and phosphates. They make up the outside of the molecule. Deoxyribose is a pentagon shape, and the phosphate is just a small molecule in between. Next, we need to remember a rhyme, AT2CG3. So the nucleotides with 2 hydrogen bonds must be A and T, and the ones with 3 are C and G. To tell which one is which, you need to know that pyrimidines have 1 ring and purines have 2. So cytosine and thymine are the pyrimidines, so they're the 1-ring bases. Adenine and guanine are the 2-ringed bases. And now you have a labeled DNA molecule. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at SciencePet.
Okay, so that gives you a good overview of DNA and its general structure. So we want to talk for just a minute about the 2 people that actually actually the 4 people that came up with the structure. This took a lot of work. This was back in the early 50s, and there were several scientists trying to work on figuring out what it looked like. They didn't know if it was a single helix, double helix. Some even thought it might have been a triple helix. It was unknown at this time, but many scientists had been working on it, trying to put little paper and cardboard models together to try to understand what its structure was. Now, this lady right here is Rosalind Franklin. She was a scientist, but unfortunately, particularly back in the 50s, women in science were not taken very seriously. And even though she played a huge role in discovering the structure of DNA, because she was a woman, her ideas were not taken seriously, and unfortunately, she passed away before she was actually given the award. The picture that you see here is an X-ray that she took of virus molecules of DNA found in viruses. And again, they were trying to find the structure of DNA, and one thing that she had come up with and others were to take pieces of DNA and freeze them solid and then slice them and then use a special X-ray machine to take a picture of them looking down lengthwise. And as you can see right here, it forms sort of an X-looking structure. Now, it may not look like much to you and me, but to those that were trying to figure out the structure of DNA, this little X structure showed them for sure that it was a double helix. And so Rosalind Franklin played a huge role in this. Now, these 2 men right here are Watson and Crick, and they're the ones that actually won the Nobel Prize and are now considered to be the 2 people that discovered the structure of DNA. Keep in mind that there were others involved, including Rosalind Franklin. The story goes that Rosalind Franklin, her pictures got into the hands of these 2 men after she left the university where they were all working. And when they saw her pictures, they knew the structure then and there, but she never really received any recognition for her work. So it's unfortunate that this happened, but thanks to these 4 people, they discovered the structure of the most important molecule on the Earth, and that is the structure of DNA. And today we, of course, call it a double helix. It is like a twisted staircase. DNA stands for deoxyribonucleic acid. As it said in the video, deoxy stands for no oxygen. Ribo is the type of sugar that it has, which is called deoxyribose. And nucleic means it's found in the nucleus of eukaryotic cells, and it happens to be a weak acid. It is called the master molecule for many reasons, but it ultimately makes you who you are and codes for all the protein and materials that your body uses on a day-to-day basis. Everything is encoded in your DNA. Enzymes are very important because they play a role in actually controlling the chemical reactions that happen in our body. And DNA makes these enzymes, and the enzymes not only do that, but they also control DNA making proteins, DNA copying itself, and the entire function of our bodies and everything within it is controlled by enzymes. So within the structure of DNA is found the instructions and the information for life. And think about it this way: when you have an unborn baby, when egg and sperm unite to form a new cell, the DNA instructions know how to create the proteins and materials needed to create a new life. And at the very beginning, when we're inside of our mothers, think of it as like all of the instructions for DNA are actually turned on. And then as our bodies are made, our brains, our skin, our bones, our hair, when these things are made, think of it as like switches. All the switches are turned on at the beginning of life, but as these things are being made, many switches turn off. That's why if we damage our brain, for instance, even though DNA made our brain once before, those switches are turned off. And so once the damage is done, the damage is permanent. There's really no way to make those switches turn on yet, I should say. We are learning how to activate some of these switches that tell DNA to do things. But ultimately, once most of who we are is formed, the main function of DNA is to make proteins, because our bodies are mostly protein. We need it for repair. We need it to make blood, hair, skin, fingernails, to maintain our organs and the important molecules within our bodies. DNA itself, the actual structure of DNA is made up of subunits called nucleotides. A nucleotide, as you see here, is made up of 3 main parts: a phosphate group on the outside, a sugar in the middle, which is called deoxyribose, and then a nitrogenous base in the center. And those nitrogenous bases are what binds one side of the helix to the other, and they are A, T, C, and G. So please remember that there are 3 parts of a nucleotide. You're supposed to draw one of these, so I will pause for a moment to allow you to make a simple drawing of this. Okay, we will now continue. So the next thing is we're going to talk about those 4 nitrogenous bases. These are the things that hold the 2 sides of DNA together. They are adenine, which we abbreviate A, guanine, which we abbreviate G, cytosine, which we abbreviate C, and thymine, which we abbreviate T. So remember A, G, C, and T. They bond together like this. A always goes with T, and G always goes with C. A good way to remember this, actually I'll show you that in a minute. Another thing I want you to understand here is the amount of A, for instance, will always equal the amount of G. The amount of T will always equal the amount of A. So they always go together. I think I said that wrong. Let me do that again. A goes with T. So if there's 20% A, there's 20% T. If there's 40% G, there's 40% C. They always go together. So a little bit of fact information here. If you could take a piece of human DNA, which we actually can do, and unravel it, completely untwist it into a long straight chain, it would be over 6 feet long. And if you took all the DNA in your body and put it end to end, it would go to the sun and back 600 times. That's how much DNA there is in your body. In our bodies, because we're eukaryotic, DNA is always found in the nuclei of our cells. If you're a bacteria, it's just found free floating around in the cytoplasm. DNA is the blueprint of life. So again, A goes with T, C goes with G. We're now going to learn what's called Chargaff's rules. Chargaff's rules, basically, they're named after a guy by the name of Erwin Chargaff, who was a chemist, and he figured out that the ratios of A and T and C and G were always the same. And this is very important. So the way I came up with to remember this is, remember apples grow on trees and cars go in the garage. I'll say it again. Apples grow on trees and cars go in the garage. That's very easy to remember. Apples grow on trees, cars go in the garage. So here's an example. On this right here, I have given you 22% A. So if you have 22% A, how much T do you have? So if you remember Chargaff's rules, A goes with T, there has to be 22% T. Now that we know A and T, how do we find out how much G and C there is? The easiest way to do this is actually to subtract one of those numbers from 50, because you have 50% A and T and 50% G and C. So if you take 50 minus 22, you get 28. So that means G and C are both 28. So the answer to this is A is 22, T is 22, G is 28, and C is 28. There is another example for you to do on your own. Please do that now, and I will pause. Okay, to continue for just a moment, a good way to think of the way DNA is held together is more to think of it as like 2 sides of a zipper. Think of a coat that you have. You hook the zipper together and then zip it up to make one zipper, but it's really made up of 2 parts. DNA is exactly like that. You have one part on one side and one part on the other, and the zipper is held together with what are called hydrogen bonds. They're weak hydrogen bonds. They're designed to literally unzip. Just like it's usually easy to unzip a jacket, DNA can also be easily unzipped using enzymes. So to help you understand what the structure of DNA looks like for real, if you think of a chromosome, chromosomes look like these X-looking things, as you can see in this picture. They look like X's. But what it really is just a piece of DNA that's tightly wound together. It winds very, very, very tight, and if you can see sort of in the middle there, it's wound around these little ball-like structures called histones, and then it twists and coils until eventually, when it's all coiled up, it forms a chromosome, which are kind of shaped like an X. When it unravels, it forms the double helix chain. So the sequence of nucleotides forms the unique genetic information of an organism. The closer the relationship between organisms, the more similar their DNA nucleotide sequences will be. Okay, and a good way to think about that is how closely are we related to a chimpanzee, and how do we know? We know that we share 98.6% of the same nucleotide sequences as a chimpanzee. This makes complete sense because a chimpanzee is very much like us. They look a lot like us. They have the same hands, the head's in the same place, their ears and eyes. Even though they're not as intelligent as we are, they still have the same general body structure and shape, and so we're very closely related to them. But think about this. I have a question on the bottom that says, are you more closely related to an earthworm or a tree? Believe it or not, we are related to trees, and we're related to grass and plants. We share DNA with them as well. To make a tree, you need DNA. To make an earthworm, you need DNA. But if you can think, we actually are more closely related to an earthworm than a tree, because an earthworm is at least an animal, and it does have a primitive brain and blood and some blood vessels and muscles, where trees don't have any of those things. So our DNA is going to be a lot less like a tree than it is an earthworm. So to understand the next part of this is, how does DNA actually make a copy of itself? Whenever a cell divides through mitosis, which we've already talked about, DNA has to make an exact copy of itself. It's done in S phase of interphase, as we've already talked about. The process where DNA makes a copy of itself is called DNA replication. I've got a short video on how this happens.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, The enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Isn't that an amazing process, and that we understand it? We understand it so well that today we can use technology where we can actually go in and do repair and change sequences in DNA. We do it all the time. With the coronavirus vaccine, all they did was they were able to take the actual DNA from the virus itself, and they figured out which part of it made a protein on the surface of the virus, and then they simply, I shouldn't say simply, it was very complex. They used DNA to create a 3D model of that protein and then found the code for it. And when we get the vaccine, all they're injecting us with is the code and nothing more, just the code to make that protein on the surface of the virus. So in our muscles, our body goes through a process where it converts that code into and replicates the protein fragment that's found on the surface of the virus. And our body recognizes that, and so if we do come in contact with the virus, in theory, we are protected because the body has already recognized that protein on the surface. It's a brilliant, brilliant technology, and to do it in a year was amazing. And it's going to open the door to make new vaccines and new medicines much faster than we have in the past. And this was all done just by a handful of scientists that figured this out, studying other coronaviruses for the last 20 years. It's absolutely amazing. So just in conclusion, as we finish up, make sure you understand that DNA, when it copies itself, half of the strand is the original from the original strand, and then the other half is newly replicated. So it's called semi-conservative. It's half original strand, half new strand. This all happens during S phase of interphase, as I already said. It's part of that process that DNA, that a cell will go through prior to it going through mitosis and meiosis, which we haven't talked about yet, but we will. So, just a few questions to finish this up. Which of the following is not a component of DNA? So, did we talk about simple sugars, phosphate groups, nitrogenous bases, and proteins? So the only three that are components of DNA is the simple sugar, phosphate, and the nitrogenous base. A protein is not part of DNA. Instead, DNA is used to make proteins. Which of the following correctly comprises a complementary base pair? So once again, apples grow on trees, cars go in the garage. So the only one here, it says, which is the follows correctly? So the answer is A, apples grow on trees. This ends this assignment. You'll now do a practice exercise to see if you understood the concepts.